Every year, the Department of Defense spends $585 million on lower musculoskeletal injuries. The number one injury sustained during basic combat training are tibial stress fractures, also known as, or more commonly known as, shin splints. This causes people to have to restart basic training or be discharged completely from the military. Nowadays, people are more sedentary before entering into the military, and this intense amount of physical stress that's being placed on the body is causing many, many injuries and costing the Department of Defense a lot of money. So currently, there's no way to measure the amount of load being placed on the tibia during these rehabilitation plans. IMUs, or inertial measurement units, are small wearable devices, as you can see in figure 1 up there, that measure acceleration. This acceleration can then be determined to follow the load being placed on the tibia if placed in the proper position. The purpose of this study is to determine the validity of these IMUs during walking, running, and rucking. And rucking is walking or marching with a weighted backpack, and measuring the biomechanical loading that is placed on the tibia. This experiment is taking place at the Sports Medicine Research Institute here at Kentucky. We actually have 9 subjects that are completely through testing, but only 7 subjects have been processed. So these are physically active individuals with reported experience rucking and running and no reported pain. It's a cross-sectional comparative design study. These IMUs are placed on the inside of the legs, on both tibias, on the low back, and the chest. This person the participant then completes the overground procedure for validity. In this procedure, the person walks, runs, and rucks in a 3D motion capture system, as you can see in this picture above, over force plates. So the person has markers on them that are anatomical and both anchor markers. And from there, we're able to create a bone model where we're able to determine the variables that we're looking for. The reason that we're doing this is because the 3D motion capture system and these force plates are valid. So we're comparing a valid measure to a measure that we're not certain about, which is the acceleration from the IMUs. From this data, we got our correlations from our results. So we were comparing the peak acceleration in one step compared to the peak vertical ground reaction forces, the peak knee forces, and the peak hip forces during that single step. In the walk, so correlations, we're looking from -1 to 1. Closer to both ends of the spectrum is a stronger correlation. For the walking, we saw weak to moderate correlations with the numbers you see in the top figure. For the running, we actually saw moderate to strong correlations between the peak acceleration and especially the peak knee forces. Rucking, we saw moderate to low. So the conclusion of this study is it is ongoing, so we don't have all of our data just yet. So from this, we can't determine that the IMUs are 100% valid in measuring exactly what we'd like so far, but we did see strong correlations in the running trial that we had, which is very good for rehabilitation plans because that's very common in tibial stress fracture rehab. You mainly focus on running for these plans. So subject testing is going to continue. The strong correlation in the run is something that it's worth looking into further, and it's promising for our study. So tibial stress fractures are such a problem in the military. It causes people, you cannot run, you cannot be physically active, and running is extremely important for our military. That's something that they're constantly doing, especially with the weighted backpack. That puts immense stress on their bodies. So currently, therapists have to use the amount of distance a patient travels during a work period in order to determine the amount of work that they're actually being done. So this isn't directly measuring the load that's being placed on the tibia. It's just kind of giving a general idea of how much work is being done during this time. So if these IMUs are determined to be valid, we could go further. And this is just looking at the peak acceleration in one step. This could lead to future studies where we could get a cumulative load, so all of the acceleration during a session, and you can determine how much force is exactly being placed on that tibia. From there, rehabilitation plans can be individualized, and this can help each soldier on a just one-on-one basis. So with further testing, IMUs truly have the potential to help our nation's military. And as a future physical therapist, I love this sort of study because you're not going to have a large room with a 3D motion capture system. You need something that's applicable to your field. Thank you.
All right, how many of you all know anything about fetal alcohol spectrum disorders or fetal alcohol syndrome? A few, all right. How many of you all know what the Surgeon General recommends about drinking while pregnant? Okay, more, that's good. So the Surgeon General recommends that there's no safe amount of alcohol to drink while pregnant. I also want to point out that fetal alcohol spectrum disorders is a spectrum disorder, just like I'm sure you're familiar with autism. So this image right here just shows that fetal alcohol spectrum disorder is an umbrella term, and fetal alcohol syndrome lies within the spectrum. So the purpose of our lab is to understand animal models of prenatal and neonatal ethanol exposure, and also to help try and reduce some of the deficits of fetal alcohol spectrum disorders. So we use several models in our lab, but for the one I'm talking about today, our hypothesis is that the flavonoid Solidago nemoralis, which I will talk about later, will reduce the deficits of fetal alcohol spectrum disorders in a rodent model. So why is our research so important? Fetal alcohol spectrum disorders are as common as ADHD, but only 80% of individuals with fetal alcohol spectrum disorders are diagnosed within their lifetime, or undiagnosed within their lifetime. A lot of times, doctors don't want to diagnose because this field of research is fairly new, and there's not a lot of treatment methods that are known about fetal alcohol spectrum disorder, so doctors don't want to make moms feel guilty for no reason, and they just won't diagnose altogether. So it's a huge health disparity in our society. So as I said before, we do use rodent models, and we recognize that rodents are not humans, but that's okay, because in this situation, it just gives us a simplistic model, and it gives us good face validity. And face validity means that we see the same deficits in the rodents as we see in humans. These deficits can range from poor balance and coordination to hyperactivity to poor decision making and planning, and memory, learning deficits, things like that. So in this study, we have 4 treatment groups. You can see these are the results. Okay, so I'll talk about the methods first, sorry. So we use 3 different tests to test this model, and we use open field was the first one, and it's where we basically put a rodent in a chamber and measured their hyperactivity for about 30 minutes. And we also measured where they spent most of their time within the chamber. And so this clues us in as to how their hyperactivity is affected by neonatal ethanol exposure. The second test that we use is water maze, and it's a spatial learning and memory task. So on day 1, they have to learn the maze, and on day 2, they have to remember what they learned from the first day. So deficits in this task might show that there are deficits in the hippocampus, which is the brain region responsible for learning and memory. Then the last task that we used was called ASST. It's a decision-making task, and so they're presented with several options, and they have to choose the correct one, and they're rewarded for it. So deficits in this task might show that there are deficits in the prefrontal cortex, which is responsible for decision-making and planning. So the compound that we're testing is called Solidago nemoralis. It's derived from the Kentucky state flower, which is the great goldenrod. And this a flavonoid just means that it has antioxidant and anti-inflammatory properties. And so these properties we're thinking are what help protect neurons in a developing brain. So in our model, we had 4 different treatment groups. We have a group that was exposed to ethanol only neonatally, a group that was exposed to Solidago nemoralis only, and then a group that was exposed to both Solidago nemoralis and ethanol, and then a non-treated control group. So these are our results from the water maze. We split it up into male and female just to make sure that there were no gender differences, and that there weren't for day 1. So what we're looking at here is on the x-axis we have day 1, which is where they learn the task, and day 2, again, which is where they had to remember the task. And basically what we found is that the group in yellow, the group that was exposed to both ethanol and Solidago nemoralis, did consistently better than the group that was exposed to ethanol only. So the lower amount of trials that took them to complete the maze, the better, is what we're getting at here. So we concluded from these results that 1 dose of Solidago nemoralis can help reduce the deficits of fetal alcohol spectrum disorders, which is huge. So if you're going to take away anything from this presentation, just take away the fact that you can advocate for this by letting people know, a woman know that if she's pregnant, you should just advise her that she should not be drinking while she's pregnant. And also, if a family thinks that their child might have a fetal alcohol spectrum disorder, they should talk to their doctor about it, because there are behavioral therapies out there and research is still being done in this field. Thank you.
I have had the amazing opportunity to spend the last 3 summers in Africa conducting research with UK. And while I'm there, you notice these huge trees called Croton Megalocarpus trees, or Croton trees. These trees can produce an inedible nut twice a year that usually just falls to the ground and is left as an organic waste. The company that we partnered with in Kenya, called EcoFuels Kenya, has developed a process to extract the oil out of this croton nut and sell it as a biofuel or a diesel fuel. The leftover seed cake is then used as animal feed for surrounding livestock in the area. This still has a waste product, though, the husk that surrounded these nuts, and using a slow pyrolysis process, we're able to thermally decompose these husks, otherwise known as just heat them up over a long period of time in the absence of oxygen, and using that we can extract volatile components out of the husks and condense them into a liquid we are calling wood vinegar. Now, this wood vinegar is mainly comprised of acetic acid and some methanol and other wood oils. Literature suggests that this wood vinegar is very effective as a pesticide, herbicide, and fungicide, and all that must be done is it must be diluted with water and sprayed on the crops. The pyrolysis process not only results in wood vinegar, but it also results in a biochar, which looks similar to charcoal. And this is very nitrogen-rich in composition and can be pulverized and added to the soil as a soil amendment to retain nutrients and water. So out of this, we get both a pesticide and a fertilizer. I spent all of last school year before we went to Kenya doing small-scale testing on the wood vinegars. So I was isolating different variables that I thought could be affecting the percent yield and the composition of the vinegar product. Isolating the operating temperature, I found that the higher the operating temperature, the better the yield we got, but we do need to be careful because while I was using electric power, developing nations use a different fuel source, usually firewood. And also, as we increase the moisture content of the husk by adding water before the pyrolysis process, the percent yield would go up, but also it would help extract more organics from the husk, so the composition of the vinegars would be better. We took this information with us this summer over to Kenya, where we conducted large-scale testing on the kilns owned and operated by EcoFuels, shown here. Now, we were testing the same parameters and variables, but also the insulation structures of the kilns and condensing procedures. So they were using both water-cooled and air-cooled. The condensing procedure didn't seem to account for much, but the temperature gradient inside each kiln, it was better when it was completely insulated, but still not what we had when in Paducah due to it being a different fuel source. We took every vinegar sample home with us, though, ran both small and large scale through a gas chromatograph machine, and showed that there is mostly acetic acid, as shown in this peak here, and methanol. Now we can know that this is acetic acid and methanol due to calibrated standards that we have run ahead of time. Going forward with this project, though, we need to figure out what is in these bottom 2 peaks because we do not have calibrated data that match those timestamps, and we also need to conduct tests on how effective this is. We currently have those ongoing both with the University of Kentucky in this state and over in Kenya. Thank you.
My project's focus was upon the Appalachian region due to its abnormally high rates of mental health disorders. In the Appalachian region, the level of alcohol abuse is far greater than the rest of the United States, and alcohol abuse has been known to lead to neurodegeneration. The Appalachian region is also afflicted by widespread heavy metal contamination brought about by the coal mining industry that has been there for decades. Heavy metals such as arsenic leak into the residents' drinking water and soil, and arsenic has been known to cause neurological damage. So my hypothesis for this was that the combination of arsenic and ethanol would cause a synergistic increase in the neuron cell death experienced by these residents. To test this, we cultured a line of neuroblastoma N2A cells and treated them for 48 hours with ethanol, arsenic, and a combination of both. We chose a concentration of 200 milligrams per deciliter for alcohol because it represents and falls in the middle of the physiological range for the blood alcohol concentration of people with alcohol abuse. We chose a concentration of 2 micromolar for arsenic because it would be found in some of the residents' wells in that area. For our first test to analyze our results, we performed an MTT assay, as seen over here, and that effectively just measures the cell viability after treatment. What we saw is that the green ethanol column had no significant difference between the white control column, and then the blue arsenic column had a significant decrease in the cell viability compared to the white control column. In the red, we see that the combination treatment had an even more significant decrease in cell viability compared to the control and the other treatment groups. What's important to note here is that although the ethanol itself did not cause any cell viability decreases, when combined with the arsenic group in the blue, we see that there is an even more significant decrease, which is interesting to note. So a decrease in cell viability tells us that cell death in some form has occurred. And to determine what type of cell death has occurred and the degree to which it occurred, we performed a flow cytometry assay. And in this, we just marked our cells with PI and annexin, and then once we received the values, we plotted them out, as seen over here, and we saw that these points that lie in the top right quadrant for each of them tell us that PI and annexin gave positive readings. And these positive readings for both of them tell us that cell apoptosis has occurred, or programmed cell death. And we can see the percentage of apoptosis cells in the bottom here. What we see is that in the ethanol green column, there is no significant changes in cell apoptosis compared to the control. In the arsenic blue column, there is a significant increase in cell apoptosis compared to the control. And in the red combination treatment column, once again, we see that there's an even more significant increase in cell apoptosis compared to the control and the other treatment groups. It's important to note here once again that ethanol itself did not cause any changes in cell apoptosis, but once combined with arsenic, we see a drastic increase. So we conclude that arsenic and ethanol synergistically act to further increase the amount of neuron cell death experienced by these residents, and that could explain for some of the abnormally high rates of mental health disorders experienced by them as well. Secondly, we note that apoptosis was the major form of cell death experienced here and caused by these two. A future line of study could be performing an animal model where we have, say, a mouse and introduce it to water treated with ethanol, arsenic, and both to get a better representation of what would be happening to these residents. Thank you.
So if I could tell you today how soon you would die, would you want to know the answer to that question? I think regardless of what your answer is to that question, medically that's a very relevant thing. We know that for smokers, oftentimes the only time they quit is when they know they have a lung cancer diagnosis, when they know they're going to die. And so I envision in medicine in the future, we would be able to accurately predict how soon people would die and potentially let them know that they need to modify their lifestyle or else they'll die soon. And that could be a motivating factor. So all of us in this room, mostly as college students, are age 18 to 22 years old. And if you're one of the judges, a little bit older. But the fact is, not all of us will live to the same age. Some of us will live to 60, some to 80, some to 100. So why is this? It's because chronological age and biological age are not the exact same thing. Chronological age simply being how many calendar years you've lived, and biological age being how old your cells are, how old your organs are. And because of this sort of mix-up between these two, we need to develop what are called biomarkers of aging. A biomarker of aging is simply an accurate thing you can take from an organism, whether it is blood or cells or whatever it may be, that tells you how soon that organism is going to die. We have some things, when you go to the doctor, you get your cholesterol, you get blood pressure, all of these things, but they don't actually accurately predict how soon you'll die. Now, I work in a lab who's very interested in metabolism here at Kentucky, and one of the proteins we've been interested in for a long time is this Chemerin protein. I'm not going to go into detail on what Chemerin does physiologically. I'm happy to talk about that in the question and answer. But what you need to know for the talk is that Chemerin is secreted from fat cells and it induces inflammation. It's a bad protein. People with cardiovascular disease, people with diabetes, smokers, Chemerin levels are all very high in these individuals. Now, I also have an interest in aging, as my talk suggests. I did an internship last summer at the National Institute on Aging in Baltimore, and I wanted to sort of combine my interest in metabolism and aging. And they had a great cohort of aging mice in Baltimore, so I set up a collaboration between our lab and the lab I was in last summer, and wanted to look if this Chemerin protein was a biomarker of aging. It increased in all these other unhealthy behaviors, obesity, smoking, diabetes. I thought quite possibly it could increase with age as well. So I hypothesized that the young cross-section of mice would have low levels of Chemerin, and the old cross-section of mice would have high levels of Chemerin. So what you can see here on the y-axis, we have Chemerin levels, and then the male and the female group, young being in red and green being in old. In the males, the young have relatively low Chemerin levels as well as the females in their blood. But in the old males, you see a significant increase in Chemerin, and also in the old females, you see a dramatic rise in Chemerin. So this data is obviously very preliminary, but there's two very important directions we're going to take. The first being, does this actually predict how soon these mice are going to die? The thing about aging research is oftentimes your mice live a really, really long time. These mice are still living up there in Baltimore, and so we don't know how many of them are dead. What we're going to have to do is determine if Chemerin is a predictor of how soon they die to see if it's a true biomarker of aging. And the second thing is to look at this in multiple species, rats, monkeys, and eventually humans, and see, is this just something happening in mice, or is it also something happening in humans? So the take home here is, one, that this is the first time that Chemerin has been shown to be a biomarker of aging, which is exciting in and of itself. But the second is why you should care about biomarkers of aging. I envision a day in the future, in 20 or 30 years, where you could go to the doctor, and you could get your cholesterol, you could get your BMI, all of these things, and you could get things like Chemerin level, or simple assays in your cells that tell you how much longer you have left to live, so that you could potentially know to change your lifestyle, to change how you age, and also we can understand more about the aging process. Thank you.
We all want healthy family relationships, but according to the data, a third of you had a distrustful relationship with your parents when you were young. In other words, you did not trust that they would meet your needs or that they would be a stable presence in your life. Within the field of psychology, we call this attachment, and it shapes everything. Specifically, that's an insecure attachment, whereas a trusting relationship is a secure one. And whether you have an insecure or a secure one shapes your relationships, it shapes your worldview, it shapes your mental health. In fact, we've known for a while that children with an insecure attachment are significantly more likely to develop depressive symptoms later. But we didn't really know why this connection was as massive as it was. So to study this, I joined Euroscholars, which is a research abroad program that UK sponsors, and I studied under Dr. He Bosmans, as I mentioned, to ask the question, why? Why are securely attached children seemingly more resilient against depression, while their insecurely attached counterparts are at greater risk? Now, if we could begin to answer that question, then maybe we could begin to interrupt this particular part of the depressive cycle. And we had a ton of variables with which to try to attempt this, right? So if you look at my slide, we had a self-report measure of depressive symptoms, and then we actually had 4 measures of attachment. Now some of you are thinking, why did you have 4 measures? And there's a good reason for it. Right now within psychology, there is not a gold standard for how to measure attachment in middle childhood in particular. So rather than engage in an intellectual kickboxing ring to duke out which was our favorite, we used all 4. And then we had hundreds of other variables to try to plug into that B slot, that middle slot, and see what could be contributing to this relationship. And a surprising variable we tested was gratitude. Gratitude is a surprisingly complicated construct, but it's essentially a positive response upon receiving something. And when someone experiences that all the time, we say that they're a grateful person or that they have a positive orientation towards the world. Now, if you look at my slide, you can kind of see why gratitude fits into the same world as attachment and depression. We believed that children with a secure attachment were going to be more grateful later in life, and then that was going to help protect them against developing depressive symptoms. And after I performed dozens of analyses and also looked at alternate models so that I could rule them out, for example, this same model but backwards, the data confirmed that this does appear to be the case. And that's exciting for a whole host of reasons. We were the first team to look at this relationship, but more than that, this data spanned 3 years. So it's really exciting to find data that's this robust over that long of a period, especially when most developmental research tests everything in one shot. But why, you ask? Why were the results so robust, and how is this supported by theory? Here's essentially what we believe is happening. When a child is developing an attachment with a parent at an early age, they're expressing needs. But then the parent is meeting those needs, and that generates gratitude. As this happens over and over, that gratitude, we believe, can crystallize into a trait or a personality feature. So the parents are training the children to have a positive orientation towards the world. And children with a positive orientation towards the world are less likely to focus on the negatives and thus more resilient against developing depression. And this has massive implications. Depression is one of the most common and one of the most serious disorders in adolescence. And as we've discussed, insecure attachment is also incredibly and shockingly common. The good news is that there are very well-researched gratitude interventions or programs that are specifically designed to increase gratitude. And per this model, that could be exactly what insecurely attached children really need. What would happen if we incorporated gratitude interventions into the American foster care system? What would happen if we used them more in family-based therapies or in the elementary schools where we know that 1/3 of our students are at risk? It would seem that similar to a flu vaccine, we can help inoculate our kids against things that would harm them. Now, future research has a lot of work to do. For example, we focused on a maternal attachment, a child's attachment to the mom, but we need to look at how a dad could play into these relationships. In the meantime, we're excited about the results because knowledge is always the first step towards healing.